We wrote top 5 challenges on the board, and number 1 was all nanoparticles go to the liver. And we spent the next 5 years elucidating those rules. And now you have the blueprint how to break the rules. So now we had, for the first time, a way to deliver genetic medicines into the lungs. And this has not been seen before. There's something unique about the attributes of this delivery nanoparticle that it's now in the lungs. And those are the feelings you have when you think this could be a new therapy for somebody. This could be a new medicine.
Welcome to Learning from Leaders, a podcast from Southwestern Medical Foundation where we dive into a world of wisdom, mentorship, and innovation, exploring the insights and life lessons of seasoned community and business leaders. I'm Michael McMahon, President and CEO of Southwestern Medical Foundation. I invite you to subscribe now and join us on this empowering journey of growth and leadership. Now, let's learn from the best and drive meaningful change together.
Welcome to Learning from Leaders. I'm Mason Smith, and today we're joined by Dr. Dan Siegwart from UT Southwestern. He's a professor of biomedical engineering and biochemistry and the director of UT Southwestern's program in genetic drug engineering. Most notably, Dr. Siegwart leads the Siegwart Lab, where he and his team develop nanoparticle technologies designed to deliver genomic medicines. Dan, thank you for joining the show.
Oh, it's an honor to be here. Thank you.
Well, Dan, it's so great to have you here. I wanted to say a quick aside. Today is a really special conversation for me because I have had the privilege to get to know Dan as a friend and a mentor and a business colleague for many years now. I think we probably met in 2022, 2021. And Dan is literally one of the leaders I have learned from in my life. And so I'm really thrilled to share his work and showcase him today.
Oh, thanks. I've learned from a lot of leaders myself, and we'll talk about that today.
Perfect. Well, to jump into things, I'd love to cover what is maybe something unexpected that a lot of viewers might not know about being a scientist.
There's an enormous amount of creativity in scientific research and the scientific enterprise, and that's not commonly talked about. So myself, I just love ideas. I love thinking about ideas. I love working on ideas. They're ideas just like an individual wants to paint a painting and they have an idea of what that could look like, or they hear a noise and it gives them an inspiration for a new song, and that's an idea, and they work on that song. I think scientists come up with these ideas and they come from a lot of different places. It's just really, really fun to be a scientist and work on ideas that are interesting to yourself. I think some scientists are termed fundamental scientists. It means that they are studying the basic processes by which the earth and human bodies work. So, for example, someone might have a burn on their skin, and this burn does not always heal correctly to look like the skin from before. So fundamental scientists could look at that and study, okay, what is the composition of skin? What are the factors that make the skin regrow or not regrow? How do scars form? And what are the molecular mechanisms by which these processes take place? And that understanding is just fundamentally interesting. And it can also lead to that investigator or potentially others, more likely others, coming up with new strategies that might make that burn heal much more effectively. Other scientists are problem solvers. These individuals identify a good problem, and that's a very purposefully vague term. But a good problem means that if you solve it, there will immediately be 1, 2, 3, 4 obvious, broader, high impacts that result from that problem being solved. And that's how you know that you're on to something that's a good problem. That falls into this area of applied science, whereby scientists will apply the basic knowledge to solve a fundamental problem. I think there's probably a third category, which is creativity or curiosity. As a chemist, that's my background. There is a phrase I use all the time, which is chemical curiosity. And there are molecules that never existed on Earth before, and yet we can conceptualize them and make those molecules in the lab. And sometimes they have very unique properties because they never existed before. And sometimes those unique properties can affect a fundamental goal or an applied goal. Yeah, so that's what I love about science, is coming up with ideas and being able to realize those ideas in a meaningful way in the laboratory.
Yeah, that's such a great framing, the importance importance of creativity in science, and maybe we can jump into some of that applied creativity that you're talking about in your lab. I'd love to unpack a little bit, especially for a viewer that's a non-scientist. What is a nanoparticle? What is a genomic medicine? These are the things you work on in your lab, but how would you explain your work to someone that is non-technical?
Yeah, nanoparticles are small spheres, fundamentally. Small meaning they're smaller than the width of the human hair. That means we cannot really see them. But if they were to be dissolved or dispersed in this cup of water, it would look cloudy and usually blue. And that blue is due to how the light is reflecting off of those very small nanoparticles. And we have techniques to characterize exactly what those nanoparticles are made. But for a lay audience, they are small balls that can transport drugs inside of cells in our bodies in a very meaningful and important way. The nanoparticles that we focus on are called lipid nanoparticles, meaning they're made of lipids. Lipids are in all of our cell membranes, and it's a fundamental compound in life. My friend Katie Whitehead describes them as little balls of fat, which I really like as a phrase because ultimately that's what they're made of.
Awesome. And maybe you could explain a little bit of the why now for nanoparticles and lipid nanoparticles specifically. A lot of viewers might not know, but lipid nanoparticles are very tied to, I guess it's a little bit of past history now, but COVID vaccines, but it's now you're evolving that technology in a big way. So would love to cover the why now.
Sure. Most of the drugs that we take, that aspirin, Tylenol, ibuprofen, even anti-cancer drugs and others, are what are called small molecules. So small molecule means they're small. Obviously, it's in the name. Molecule means it's a chemical molecule. Because that chemical molecule is small, we can eat it. It goes into our stomach, gets into our blood, and basically goes all over our bodies. So 30 minutes after eating this medicine, our headache can go away. And that it remains about 2/3 of all of the FDA-approved drugs are small molecules today. What those small molecules do is they will find something called a protein, and that protein may be causing or be otherwise related to the ailment, like a headache or a genetic disease. And that protein, if it causes that effect or is somehow related to that effect, the small molecule can bind that protein and inhibit its function. It's like throwing a wrench into a spinning wheel. It breaks the function, even though that wheel still exists. And consequently, your headache goes down, or the symptoms of your disease go down, or maybe the cancer cell dies. Now, this remains a great way to develop medicines. And in the spectrum of what we understand as the central dogma of biology, every one of our cells contains DNA. DNA is the blueprint of life. Periodically, at different times, for different reasons, cells will transcribe the DNA into a messenger molecule called messenger RNA. And that messenger RNA is now in the cytoplasm, the soup of the cell, where it's translated into protein based on the genetic sequence. And these proteins are doing everything in our bodies, from forming the structures of our cells and our existence to communicating and orchestrating these complex signaling pathways that make humans humans. So genetic medicine is essentially saying, yes, we can take those small molecules that can bind and inhibit a protein, but potentially we could go upstream in the central dogma of biology. We could, for example, design a molecule like a short interfering RNA that will bind to the messenger RNA and lead to destruction of that molecule. So now if there's no messenger RNA, there's no protein. And if that protein was causing somebody's disease, it's gone now. We could also go upstream from there and use something called genome editing, which could fix the mutation that's causing the mutated mRNA and the mutated protein that causes somebody's disease or other ailment. Even cancer could be targeted this way. You could go into the DNA of the cancer and inhibit that molecule in a way that the cancer cell could no longer survive. So genetic medicine is coming alive because the tools and knowledge and technologies exist to design specific, safe, and effective strategies to work upstream of those proteins for the first time. And it's becoming a larger class of medicines, including 8 siRNA drugs that are approved and hopefully many more that inhibit the mRNA step or the DNA step to really provide long-lasting, durable therapies for people.
Yeah, I think what would be super helpful is maybe a compare contrast. I think a lot of people have heard of CRISPR and the discoveries by Jennifer Doudna. How does that compare to the work you do? Which your work is more, it's on the delivery side, but it's also on the mRNA and the RNA level. So the transcription layer, that middle layer of the central dogma, I think that would be really helpful for the audience.
Right. Well, the complexity of this small molecule protein drug interaction is hard to find those perfectly sized molecules that can fit the protein. From a drug design point of view, the intervention at this earlier stage is a little bit easier because we know the sequences of the mRNA or the DNA. But those medicines, unlike the small molecule, as I described, small molecule can go everywhere if it's designed properly. These RNA drugs or DNA drugs, they cannot cross cell membranes on their own. They are negatively charged large molecules that just cannot fit through the membranes of our cells. So that necessitates a delivery solution, which is where we come in. So we use these lipid-based spheres to encapsulate those RNA or DNA drugs. Those molecules are then protected from degradation, and due to the properties of the nanoparticle, it can mediate entry into our cells and release the nucleic acid so it's functional in the cytoplasm or in the nucleus to tie up the earlier description of that process. So delivery is an essential or required part of the development of genetic medicines. But if you get that right and you have a strategy that's safe, and I think today we have a safe strategy for the liver of human beings, we have a safe strategy for vaccines, we potentially have safe strategies for other tissues that's definitely emerging. And with that backdrop, you can now design the messenger RNA to make virtually any protein. You could design something like CRISPR that Doudna and Charpentier discovered that can go site-specifically into the blueprint and, say, fix something that went wrong after birth. Yeah, it's a really exciting time, I would say.
Definitely. I think you and me could probably go on and on about the specifics on the science, and we definitely will dive into that more later on in the conversation. But I'd love to maybe take a step back and circle back a little bit to your background. And since this podcast is the Learning from Leaders podcast, you've studied under some amazing leaders in the labs you've worked under at places like Eli Lilly and Carnegie Mellon, University of Tokyo, and MIT, and maybe most notably at MIT under Dr. Bob Langer, who's a very luminary in the field. Can you speak to the various mentors you've had, the leaders you've had in your life?
Of course. I've been fortunate to be able to study with and learn from some remarkable leaders in my time. And I'm laughing about it right now because the whole thing is remarkable. I was born son of a construction worker outside of Pittsburgh and didn't really know much outside of my neighborhood. There were very few role models growing up that I could learn from and lean on. So every time I get to middle school or high school, it's like, that's interesting, or, oh, I didn't know that a job existed. And it definitely was not until college that I realized that the job I have now as a professor leading a research team existed. And in the era of today, with social media and communication improved so much, I recognize that's a pretty ridiculous comment. But going back 30 years, I really had no idea that these jobs really existed. So each part of it was a stepping stone, and maybe thinking about it alive in the context of this podcast, perhaps my ignorance played a role in the reception that I took to learning from these different leaders, that because I did not know, I was even more eager to understand how do they command leadership in their different environments and how do they manage teams. So tracing it through, I first studied with a Polish-American scientist named Chris Matyaszewski, a professor at Carnegie Mellon University. And Chris was just one of the most brilliant people I had ever met. He had such a command for the knowledge and the literature, and he was able to, on a piece of paper with us in the office, map out the beginning and the end of a project. And I would take those instructions, go into the laboratory, and do the chemical synthesis in the hoods, and more or less it all worked out. He was a very detail-oriented type of leader that functioned well being in the office and orchestrating the research of the lab. I spent about 4 months in Tokyo, where I trained with Kazunori Kataoka, a professor at the University of Tokyo. And Kazunori was more of kind of a supportive mentor. So in Japan, there is a group-oriented thinking process that I did all of my experiments in Pittsburgh at Carnegie Mellon by myself, and every experiment in Japan I did with 2, 3, 4 other people. It was much more of a team, group-oriented way to conduct scientific research. So in that backdrop, Professor Kadoka provided a role to be a supportive member of the team. So he would uplift and encourage the collaboration of that team and was generally complimentary. Bob Langer at MIT is, I think, one of history's greatest inventors, for sure. And Bob was always focused on solving problems. So he has a unique ability to get his lab members excited about the challenge of solving a problem at any cost by any method that might be necessary to solve that problem. And Bob led, I think, a bit more by example and motivation and setting the tone for the team to also collaborate together, and was somehow between these different mentors.
Awesome.
Yeah.
And I know Bob Langer is also very focused on commercialization opportunities that sprout from his lab's work. And so I'd love to maybe kind of transition towards that, because you're actually now a commercialization expert in your own right, and perhaps learning from Dr. Langer. You've started ReCode Therapeutics and also SignifyBio. So maybe if you could kind of connect the through line from your work in the Siegwart lab to those commercialization opportunities, that would be really interesting.
Yeah, but Bob is one of the true out-of-the-box thinkers that I've ever met before. And with that type of thinking, it just feels like anything is possible. So if we contemplate the mission of University of Texas Southwestern Medical Center, ultimately, I have a job as a scientist, but this job is in the environment of hospitals and patient care. So some of our colleagues are doctors, nurses, administrators, running the testing, doing all kind of amazing things that are directly relating to the healthcare of that individual. For me, I don't see patients, but I feel a responsibility to play a role in contributing basic knowledge or applied science that could improve the lives of others, of humanity. I know that's a mission, too, of the Southwestern Medical Foundation, to support and enrich those activities to be maximized in our region. So with that backdrop, whenever we have this opportunity to discover something that could potentially be a new therapy, it creates the next steps of identifying where does that capital come from, where do the teams come from. And Bob Langer has always inspired us to solve big problems, not to be afraid of those big problems, and to feel comfortable that if man can do it, I can do it type of an attitude. So with ReCode, the origin of this company arose because these lipid-based nanoparticle that can transport these genetic drugs into cells, it does look and behave like one of our natural nanoparticles that's in our blood right now, something called VLDL. It's a part of when we eat greasy hamburgers and those fat molecules get out of our stomach and are repackaged with cholesterols. They get into the liver for recycling and redeposition throughout the body, where 30 to 40% of our cell membranes are cholesterol. So because of that similarity, lipid nanoparticles today are excellent at delivering genetic medicines to the liver. That's great news for liver diseases, and some of these are in phase 3 clinical trials now. But it's not so great news for diseases that exist in all different parts of the body. So coming back to this fundamental and applied science, when I joined UT Southwestern, we wrote top 5 challenges on the board, and number 1 was all nanoparticles go to the liver. If we could unlock that, one could make a medicine for a lung disease or a disease in the pancreas or a disease in the brain, for example. So we just thought about it, and again, going back to the beginning of the podcast, we did not have any ideas because nothing we had ever tested, and we were testing thousands of nanoparticles per year, ever went anywhere else except for the liver. So you go back to the fundamentals and the first principles. So why do nanoparticles go to the liver to begin with? And we spent the next 5 years elucidating those rules, so the design rules of what are the factors that drive those things from a chemical perspective, engineering perspective, and physiology perspective. Long story short, we elucidated those rules, and now you have the blueprint how to break the rules. So once you know what the rules are, you can identify or come up with ideas of how we could break the rules. So we broke the rules by adding additional molecules into the nanoparticle that now help the system avoid the liver and go to other places in the body, including to the lungs. So now we had, for the first time, a way to deliver genetic medicines into the lungs and data and images that we could bring to OrbiMed in New York and show them this has not been seen before. There's something unique about the attributes of this delivery nanoparticle that it's now in the lungs. And those are the feelings you have when you think this could be a new therapy for somebody. This could be a new medicine. That led to the formation of ReCode Therapeutics, which is now using those lung targeting lipid nanoparticles in 2 phase 1 clinical trials for genetic respiratory diseases. Very exciting.
Learning from Leaders, along with Southwestern Medical Foundation, is powered by generosity. Your support can help fuel breakthroughs in medical research, provide critical patient care, and support medical education. Additionally, it helps support the Learning from Leaders podcast and content like this. Every gift matters. Consider making a contribution today at swmedical.org/give. That's swmedical.org/give.
Could you maybe talk about just the timeline of starting that and then where ReCode is now? It has been a decently long journey, but also incredible progress made.
Yeah, we started originally as a transfer RNA company, and for the sake of time, I'll just describe that at the time of conception of ReCode in 2015 was a very new idea. And today there's 5 tRNA companies, and I just read the first clinical trial will start with a tRNA by a company called Alterna. But in 2015, this was a very new idea. It had never been pursued as a drug before. There were questions from potential investors around the manufacturing and delivery of these relatively complex biomolecules. And so that ultimately was difficult to raise capital for as a new drug product due to the inherent risk in doing something totally new. I think time goes on, technologies mature, and now it's quite possible to do that. But 10 years ago, it was quite challenging. And with this discovery of the organ-targeted, we call it selective organ targeting or SORT, with the discovery of that process, it now opened up new opportunity to change the payload to a messenger RNA, simplify the journey of making that protein that the body's missing, and using the particle that can go into the lung cells safely. That set up a new opportunity, and investors were much more receptive to that opportunity because it was differentiated and a little closer to clinical trials than perhaps the first idea that we started on.
Fantastic. Well, I think what would now be helpful is you definitely have a lot of lessons learned from the ReCode experience, and now you've recently been working on this new venture, SignifyBio, and I'd love to learn about how you're applying the various lessons learned, especially on the leadership front, and maybe touch on different technology aspects as well, but that would be super interesting to hear about.
Sure. SignifyBio is attempting to create medicines for the masses, meaning genetic medicines were traditionally designed for rare diseases, which remains incredibly important and requires funding and potentially some private funding and philanthropy funding to ensure that everyone in America who has a disease and an applicable drug can receive it. In other words, some of these medicines are quite expensive due to the small patient numbers. But it occurred to us that we may be able to take a similar platform and create medicines that could tackle chronic kidney disease or inflammatory diseases or cancers in new ways that are top killers of Americans. And the general idea was during COVID-19, we went to the pharmacy and received an intramuscular injection. And this strategy then produces a tiny amount of a spike protein that's benign, but it does train our bodies to look for something that resembles that in the future, like a real viral infection. But with this pathway, and similarly, there are now weight loss drugs and obesity drugs called GLP-1, and those are administered subcutaneously. So we were thinking, for complex biologics, a third of all new drugs, what if we just create those medicines directly in the human body? This could save a tremendous cost because instead of making that protein drug in expensive bioreactors, you could make it directly a customized version in your own body. and would not have to ship it around either. It would be made directly in the body. So what's missing there, though, is in the context of these other examples where one is delivering genetic medicines to the liver, those medicines stay in the liver, and that's a natural process by which this biology works out. But here, with the concept of Signify, we needed a medicine that would leave the subcutaneous fat cells or leave the muscle or leave the liver and be able to find and treat that disease wherever it might be, like in the kidneys. So that comes back to basic biology, and nature has already solved this problem. Nature uses something called a signal peptide, which is like a zip code of instructions incorporated into those genes. And those instructions will tell the cell to act on the information that it's received and send that foreign protein out into the bloodstream or to the cell membrane or to the nucleus in the middle or to the mitochondria or peroxisome. And this also is just coming back, again, I just love ideas so much. When I worked with Bob Langer at MIT, we were doing these siRNA gene silencing experiments, which I know is complicated, but basically it's a lipid fat ball with a RNA molecule that can go into the liver of animals, break the messenger RNA, and now the protein is no longer made. So I could take a little bit of blood from this cheek of the animal and measure that protein concentration, which as a chemist was quite weird because all the action had taken place in the liver. But here I was able to take a little bit of blood from the cheek and measure the concentration of that protein. So moving to Dallas, we studied how is that possible? And that's because of these signal peptides. And we started to compare and contrast and cut and paste all of the different signal peptides to develop a modular platform by which we can now use a safe strategy to administer subcutaneously, just like a GLP-1 drug, and the body will produce and make that protein for months with a single administration. I think this could be life-changing for folks who have things like chronic kidney disease or enzyme replacement therapy, where they're going to the outpatient clinic a couple of times a week. We imagine in time, if this is successfully translated by Signify and brought into the marketplace, this could be a therapy that is much more patient compliant, more effective, safer, lasting longer, and hopefully cheaper.
And I think it's really important to highlight this technology can be applied to a huge array of diseases, which is another incredibly promising aspect of it. And obviously you have to pick a certain subset of that to focus on first. But if all goes well, there could be a lot of applications of this. Yet one other area I would love to explore next is kind of furthering this conversation on ReCode and Signify, collaborations with different investors, as you've alluded to with OrbiMed, but also industry and biopharma collaborators is really important. The Siegwart Lab directly has a collaboration on the research side with Pfizer, and then your companies have various relationships with Pfizer and Eli Lilly and Danaher and others. Could you touch on these relationships and how important this spectrum of collaboration is?
Yeah. For the listeners, a faculty position at a medical school like the one that I have, it is a job akin to running a small business. I'm responsible to raise all the money, hire the staff, direct the staff, write the findings up so we can share it with the public, potentially work on patents and teach classes, grade exams, and the list goes on and on. But the fundraising component of it is really critical. That's where these scientists are hired to work on these projects to begin with. And I find that it's just valuable to work with as many partners as possible on this journey. So there may be partners like the American Cancer Society that has a core interest in cancer research. And as a laboratory, we can partner with them through granting and volunteer opportunities to advance the mission of the American Cancer Society. I think other projects are more translational in nature, and there we could partner with a company like Pfizer to both educate our scientists, facilitate their degree and growth process, while at the same time enabling them to work on something that they feel confident is a real-world, valuable problem to be solved. So that's really, helped us over the years and has introduced us to a number of great leaders like Mr. Juno Kwan, who you mentioned at Actium Group. My business partner for the last 10 years is R.A. Session, born in South Dallas. And one of the things I like talking about with R.A. as a leader is he always has a plan A and a plan A prime and a plan A double prime. In other words, he can orient the team to focus on mission A as the most important thing that we've ever seen and ever worked on in our life. And it's just so imperative that we solve this particular mission and this goal of the company. And most of the time, you get there. But sometimes something happens, and that thing that happens could be out of your control. It could be economic pressures. It could be something just didn't work the way that it worked in the earlier studies scientifically. And in that case, there is a point where you have to say that this is not going to work. And RA has a beautiful ability to immediately pivot to plan A prime with the same passion that he had about plan A, meaning as somebody who works for him or with him, you have this feeling like we're always guiding towards a common goal. And most of the time you make it. But in the rare but sometimes frequent instances where it's just not possible, RA is equally excited, if not more excited, about plan A prime and describes it in a way that was the plan all along. So I think I definitely have integrated those sort of things into my own management style of my team, which is composed of graduate students, postdoctoral fellows, research technicians, and faculty staff now, where every person should be clear what their goal and mission is, what their project is, what the paper is going to look like that they hope to write. And we're going 100 miles per hour towards that goal. But sometimes nature introduces curveballs. Sometimes there's new ideas that arise that allow us to just drive the car a little bit into a different direction. And that new direction might be more exciting than the first direction. And have a lot of joy of being on that journey with everybody that I get to work with. And keep in mind these lessons that we've learned about having individuals feel excited and empowered by the work that they're doing, goal-oriented for sure, but also open to change when necessary.
Right. I think that combination of like extremely mission and goal-driven, but with really strong adaptability is something that we all can take away and even if we're not scientists in all aspects of our lives. So thanks for sharing that. I'd like to maybe double down a bit on something you mentioned. Your CEO of Signify and business partner RA Session is from Dallas, and you might be a Dallas import, but we now call you a proud Dallasite, and Signify is headquartered here. Can you touch on the importance of trying to be a leader in the biotech field in our great city?
Yeah, we were chatting a little bit earlier that I'm a huge Pittsburgh Steelers fan. I grew up in the Pittsburgh area.
We'll forgive you for that.
Yeah, you'll forgive me for that. But I was born in McGee Women's Hospital, which is right next to the University of Pittsburgh, and that's the hospital where they're putting the terrible towels on the babies as they come out of the womb. And just because you're anointed at the first day of your birth, you're stuck with that team, or you get the joy of participating on that team in a sense as a viewer. So I've really enjoyed learning from Mike Tomlin as a football coach. I think a tremendous leader who is able to simultaneously be a player's coach, wherein he can directly relate to the individuals who are on his team. At the same time, it's clear where the lines are. It's clear that he's the boss, and there are certain expectations that he preaches to his team that are the minimum and expected things that people should be doing, including like being better next year than you were this current year and using the off season to work on something that could be improved. So each year you're better and better at your particular craft. And the same thing is true in science, where I challenge our team to identify areas for improvement and consciously work on that. So each year we are a better scientist and better equipped to be successful in life. So Pittsburgh now has homegrown coach Mike McCarthy from Greenfield. We're also really excited about that. But anyway, back to Dallas. I do really love Dallas. I love living here. I'm staying here. And it's really valuable to contribute things that could benefit our community and beyond. So I'm proud that ReCode started here. I'm proud that Signify is here, is using lab space facilitated by the biotech ecosystem that's burgeoning in Dallas, and I definitely feel a commitment and responsibility, just as those coaches do that I've described, to make this place and our community better and better.
Yeah, and could you maybe touch on maybe the strengths and areas for improvement in the Dallas biotech ecosystem? I think it's probably not known for that, but there are some really promising momentum going on. So I would love to hear about that.
Yeah, I feel our community has solved some of the difficult to solve challenges, meaning where does the science come from? There are great universities and great scientists here, and great entrepreneurs here. Where does the infrastructure come from that takes capital? There's now infrastructure, lab space to rent. Where does the community come from? That's also coming along, where there are commonly held events periodically to interface with other business leaders in the community. So really an area for improvement that I hope will happen over time is there can be a series of success stories or a continuation of success stories here. And then those individuals can become the C-suite leaders that catalyze and build and grow the next sets of companies. So we can further the momentum of what we've already been able to build in the Dallas-Fort Worth metropolitan area by facilitating those leaders to continue this pipeline and bring along the next sets of leaders in their series of companies.
And very likely a lot of those leaders might come from your lab. But
Hope
and
so.
I thought it might also be helpful to highlight RA Session. He formerly worked for Reata Pharmaceuticals in Dallas, and he's had a variety of really esteemed roles at other companies as well. But RA is a shining example of that continuation of success, and I'm sure he's going to create even more from the people he trains and leads. And speaking on that leadership front, maybe let's circle back to kind of some of your leadership techniques, leadership skills that you try to utilize in your own lab, and how do you try to mentor those under you?
Yeah, again, to put in perspective for the audience, scientists do they're characterized as being introverted. We're not always introverted. I'm sort of a secret extrovert, meaning I'm not super talkative, but I'm internally very confident extrovert personality type. And so those stereotypes are not always correct, but nevertheless, we do have to put in a lot of time to solve scientific problems. This is a area of work, so to speak, that just takes time. So long hours and work is a necessary aspect of the kind of things that we do. So consequently, most of our training is in designing and executing experiments, not really in leading others or writing grants or leading teams. And consequently, that typically happens on the job, unless there's a mentor like Bob Langer who talked about leadership and strategy all the time. And I certainly do, hopefully with my own team, talk early and often about facilitating those skills that'll be useful after you've left the research lab or gone beyond the research lab environment. So from that backdrop, what was really fun but daunting was moving from MIT in Boston to UT Southwestern, getting the keys to a new lab, and that lab is completely empty. So it's very exciting, very daunting, and it was some of the best years of my life for sure, because you start thinking, okay, what are we going to work on? What are the skill sets necessary to be successful in that area? How will I behave as a leader? And personally, which could be helpful to viewers, I went back through my own history from growing up in that small region just outside of Pittsburgh to working with Chris Maniszewski and Kazunori Kataoka and Bob Langer and other people to decide what from those individuals was effective. They're all effective, but very different. What are aspects of their personalities and their leadership skills that I could integrate into my own leadership skill? Meaning, I may or may not have the same personality attributes as those leaders. And I need to determine and develop a leadership style that takes advantage of my own natural inclinations and abilities while still leveraging specific strategies from those individuals. So I ultimately decided that I wanted to be detail-oriented to a degree, like Professor Matiszewski, meaning I want everybody in our lab to know what they're working on, know what the near-term goal is and the long-term goal is, so no one feels lost and has a mission-critical um, roadmap in front of them. At the same time, what I really enjoyed in the Langer lab was this element of freedom and creativity, that I felt free to not only be successful, but also to make mistakes and learn from mistakes. That this was an environment where if you have the idea and you are outgoing enough and adventurous enough to solve your own problems, get expertise where you need it, and put the whole thing together, wow, it was a playground and dream come true for me to work there. So for me, I'm trying to somehow be in between these different experiences to leverage my own natural abilities as a communicator, where there's enough detail that lab members hopefully feel that they always know where their project is going, but still enough support and freedom so they don't feel the pressure of I cannot make a mistake. Because once in a while, those mistakes are good things, and those accidents become discoveries, and those discoveries become something that could be really valuable in drug development someday later on.
Fantastic. And maybe a specific example on this front might be interesting. There's actually a third co-founder of SignifyBio, Dr. Lucas Farbiak, who's one of your mentees. And could you maybe touch on that relationship and maybe also touch on kind of how you think about identifying talent like Lucas and mentoring and nurturing them?
Well, first on the talent front, nowadays I use my gut. I've made a couple of mistakes in hiring over the years, and those were cases where I did rely on reference letters and phone calls with former advisors to make decisions that were technically accurate, but did not feel right in the gut. So not everybody may have that, gut sense in the world, but for me, the gut is the most powerful way to make hires. So I'm generally looking for people, whether it's in the research laboratory, new students, or even in the business community or philanthropy community, just individuals who have passion that's palpable. So in their eyes, in their body language, in the excitement that they convey in describing what it is they've done and what it is they want to do in the future, you just feel it. And those are the individuals that I enjoy working with. As Mike Tomlin says, the volunteers, not the hostages, the people who are excited about living life and being here on Earth and doing good things in the world. If you can put together a team of those personality types, as Bob Langer does really successfully, the rest of it starts to fill in the gaps, I think, to a large degree. Yeah, so that's a bit on the hiring side. And then in Bob Langer Lab, there were so many life lessons that I did not even appreciate at the time, that as I start reflecting on the 4 years that I spent in Langer Lab, these things start to become more coherent. And one of these is that Bob Langer is not necessarily appointing people in different positions. Bob Langer is creating the opportunities for people to themselves jump into. And that was not apparent when I was working there. It seemed like people were going off and doing this or having this job or this opportunity or this project by direction or fate or other reasons. But now I think what potentially is happening there is Bob is creating the open doors, and then the individuals themselves are going into those open doors. So what I mean to say is that I hope that I'm in the future and in the present able to create open doors for people to get excited about and potentially jump into. So those doors look different for different people. Some are faculty members themselves now and publishing high impact papers, and I'm so proud and happy for them. Others are working in industry, they're industry leaders, and I'm so proud and happy for them too. And others were able to retain locally, whereby Dr. Farbiak, Lucas, for example, is somebody who I just work well together with. We get along inside and outside of the lab. We see eye to eye on problem solving. We rarely, if ever, have disagreements about things. And that means that he was able to jump into an open door with his ambition and skills to become an assistant professor at UT Southwestern too. And so we work together with Samantha Chatterjee and Pratima Basic and Jing Liu to collectively manage now a lab of about 40 people. Yeah.
Such the scale you've grown it to is amazing, and both on your individual lab and then the companies outside. And I think what some listeners might not grasp as well is a lot of the foundation, especially on the commercialization side, comes from IP and your ability to protect the discoveries that you make in order for them to get into the hands of patients eventually. Could you maybe touch on how do you know when to create IP and how that factors into your decision making for your lab?
Yeah, intellectual property is important across all industries. From any product that's sold ideally has some intellectual property behind it to enable that company to take in the capital required to build the business to begin with, and then justify the capital that comes back, the revenue that comes back into the company. So same is true in the biotechnology space. I'm an employee of the University of Texas, proud to be that. And we have a fantastic technology development office that does have enough resources to facilitate patents that are owned by the University of Texas. And those patents can then be licensed out into potential startup companies or even existing companies for further development. So securing those patents is really critical for attracting the massive investment that's needed in drug development. It may cost $500 million to $2 billion just to develop a drug, and that's partly where the eventual cost of those medicines comes from. Not to mention they usually fail, and so you really have to invest in multiple drugs to find one that will be successful for that business. But definitely intellectual property is a key part of that, and something that we work hand in hand on with the university. I think Bob Langer was always looking for four things in making the decision to start a new company, and one of those four things was intellectual property. That ideally the university or company has what can be called a blocking patent, something that's broad and protective enough in the United States and across the world to prevent larger companies from simply copying that idea and bringing it faster into the marketplace through their higher resources, greater resources. Second was a big paper. Scientists do all this work in the basement, and eventually we want to share it with the world. So we write this down to the best of our abilities, and we publish this in scientific journals, which are like newspapers. And our peers review those articles to ensure the accuracy. And having a big paper, which means something that is in a glamour journal like Science or Nature, the equivalent of Dallas Morning News or New York Times, something that everybody knows, helps to create validation of that achievement as being reviewed and vetted and published in the best publications in the world. The third is typically a platform technology, something that would not be a single drug that the university could license, but something that could be a family of medicines or a general platform strategy to create tools or technologies or assays or medicines in a broad set of indications. And the third is a business expertise that there are things that I certainly have learned and am capable of achieving in the scientific realm, driven by ideas and curiosity. But there are plenty of things in the business world that are outside of my training and my expertise. And having a talented business partner with social intelligence and execution ability is that fourth piece of the puzzle that allowed Bob to start dozens of companies over the years.
Awesome. And once you have those four components and the key IP and business leaders and publications, a lot can happen. And we've been touching on your companies a lot already, and some of the technical aspects and the technologies you're bringing forward. Maybe let's look into the future. If all goes great, what could be the end impact of all the work that your lab has done, maybe in 5 to 10 years in the future?
As a personality type, to bring in the sports analogies, these athletes are training to win something like the Super Bowl or the World Series. And what's remarkable as a spectator is you see these individuals achieve that goal. Once a year, one team achieves that goal. And yet the next year, they're equally passionate about doing it again. And psychologically, you're thinking, why and how? You've already won. Like, why and how are you equally determined and irrationally passionate about doing it again? So I, as an analogy, I think many scientists, myself included, just love working. We just love ideas. It's a part of our identity as a human. And once we publish a paper, I've already moved on to the next project and the next idea. And sharing that type of thinking process with my own students as a leader is something that I enjoy doing, because it helps us to be excellent at the tasks that we're doing right now, but also equally excited about the next tasks that could come down the line. So Signify is an example of something that the company is doing all the work. I'm just there as an advisor and cheerleader. But I'm really excited about that from the perspective that that could be a way to deploy a lot of medicines that would be applicable to treat tens of millions of Americans on a significant basis. And that is very gratifying and exciting. I think looking forward, there is an emergence of these tools and technologies called genome editors, which, again, for the lay audience is simultaneously exciting and scary. That's the reality of it, that genome editing has a potential to take a child like baby KJ, who was born in Philadelphia with a rare genetic disease and would have died. And because of genome editing, that baby is alive and healthy today. The DNA that he was born with leads to a genetic disease that would be terminal, but yet you can use these molecular scissors to fix the DNA at an early age. And now, probably for the lifetime of that boy, he will be normal. So that's an incredible power and privilege and responsibility to bear for scientists and doctors who are a part of those journeys. At the same time, one can use genome editing to eradicate mosquitoes. And that might be a good idea. That might lead to real destruction of some aspects of the ecosystem that would be difficult to repair if that happens. And so I enjoy talking about it because it can convey to the audience that scientists are thinking both scientifically and ethically about very complex problems like this, and trying to decide, do the potential positives outweigh the potential negatives of something like genome editing? And where we often come to is that in some cases, just like all doctors have to do with their patients, the benefits outweigh the potential concerns or challenges of a potential procedure or drug. I think similarly, there are settings like baby KJ where this could be a life-saving medicine. And there are settings like, for example, what's called germline editing, where I think we're just not comfortable with pursuing something like that at this time. We don't know enough to confirm that's safe, and there's plenty of ethical reasons why that's not a good idea. So, just to convey that's not something that I'm working on or interested in, but at the same time, for adults who can make decisions or these rare conditions like baby KJ, which was in the newspaper, I think you can stack up enough of the positives to feel comfortable with such an approach.
Right. That makes a lot of sense. It's a very complex area of science and innovation, but there are strategies to mitigate the downsides and really take advantage of the revolutionary aspects of these medicines. One other question, a little bit of a change of subject, but it circles back to a lot of the technological techniques that you use in your lab and at your companies. It's overused topic, but it is a very hot topic. AI is on everyone's mind right now, and actually, you are a practitioner of AI in a lot of ways, and you have been for a long time. So, could you touch on that and how that's really unlocked some of the technological advancements in your lab?
Yeah, one way to frame it is that each day we have a certain number of hours that we're awake and not sleeping. And I'm trying to maximize as much into those hours as possible, both inside and outside of the lab, meaning I really do love being physically in the lab and doing that work, but I also love being at home with my family, and I'm aiming to maximize every second of every day to the best of my abilities. So in science research, if you could do one experiment per day, that's pretty good. My PhD advisor asked me to make one polymer per day, and the next day you characterize, you study everything about that polymer, and you make another one. And in 1 year, if you didn't take any days off, you would have 365 polymers. Now, if you could accelerate that by making 300 polymers per day, you would have a lot more potential for making a discovery and learning about the world or learning about problem solving. So already my lab has integrated what are called fluid handling robotics, so automated robots that can do experiments with us in our direction at a scale of hundreds per day, potentially, or thousands per week. And that definitely accelerates the timeline from having an idea to making a discovery that's useful for humanity. Similarly, I think AI has the potential and is already starting to help in that capacity by doing things like troubleshooting ideas and helping us to find better paths to solving that problem, or doing things in silico that we would normally do thousands of in the laboratory. For example, if we can narrow down in silico the investigation paths that the project might take, that definitely is going to save time and maybe open up new doors for discovery. So, a big effort of the last couple of years has been integrating wet lab data into systems that could be then prosecuted by these AI models, such that we could take our wet lab data and use AI to potentially look for things that human brain is just not able to see because of the training that we've had in textbooks and life experiences. There may be things that AI can see, new chemical compounds that would be great in drug discovery, new ways to approach problems using different materials made for another field that could be applied to biomedicine field. Those are some of the ways that it's already happening and will continue to happen.
Well, yeah, I think this is such a good example of there's a lot of doom and gloom out there about AI and perhaps negative repercussions, but this is such a key example of like how it can be used for the betterment of creation of new technologies and hopefully to impact tens of millions of patients' lives down the road. So yeah, it's I think we're going to start kind of wrapping up the conversation, but just wanted to highlight how amazing it is. You mentioned being the son of a construction worker, and now you're doing some amazing and exciting things in the biotech innovation world and in your lab. And you touched on Mike Tomlin, and
Yeah.
my hope is that maybe someday you'll be a household name like Mike Tomlin. But anyways, Dan, thank you so much for the conversation today. We did want to finish with one last segment, do a rapid fire segment just for the audience to get to know you a little better on a personal level. So are you ready for that?
Of course, yeah.
What's your go-to work soundtrack or driving soundtrack?
I do love driving. There's a lot of driving in Dallas, so most of the time I listen to the car engine and try to dissociate my day and analyze my day. But the band that I'm really into right now is called Goose, Goose. It's a fantastic modern jam band that I love listening to, and we'll be going to see them play in about a month from now in Dallas.
What is a topic or hobby totally unrelated to science that you're really into outside of work?
It might have been clear from the way I've described our scientific enterprises. Like, I just love ideas and creativity. So music, movies, writing, reading, those are the go-to creative arts that, I go to every day and really enjoy.
What's your comfort food after a long day?
I'm a son of Sicilian immigrants. I love pizza and pastas, but pizza is my hobby too. Once I've had to leave the laboratory and sit in the office, write grants. At home, I have baking steels in my oven and really love making homemade pizzas.
Awesome. What TV show are you currently watching, if you are a TV watcher?
I don't binge watch too much. I generally watch YouTube videos about cooking and race cars, and then I like all the classic movies by the world's great directors. So I like rewatching or watching the great directors at nighttime.
Yeah. If you could instantly master one skill outside of science, what would it be?
I think foreign languages, it's always something that I'm trying to learn, I want to learn, but just do not have the natural ability to quickly learn languages, unlike other fields. So if I could have a cheat code for foreign language, that's what I would pick.
And what's a fun fact about you that would surprise your fellow coworkers?
I've been blessed to have opportunities to travel over the years. First, when I was young, I did it on the cheap by literally calling friends who lived in Belgium or South America and telling them that I bought a ticket and I'm going to be at their door next week. And that, coupled to what I do now, which is travel for work, whether that's giving seminars or going to conferences, has just created an opportunity to have that privilege of seeing some of the world's great places. So in Japan, for example, my friends and I met at the base of Mount Fuji in the evening, and we climbed all night long without sleeping. And by sunrise, we were at the top of Mount Fuji and got to see the sunrise over the Japan landscape, which was pretty awesome. I've climbed along the Great Wall. I've had the chance to go to Machu Picchu and see the ancient ruins. And I just love doing that stuff because the world is such an amazing and incredible place. And being in those positions, whether that's a research lab or in the center of the former civilization of Machu Picchu, it gives you this sense of awe, and it inspires all of us to work hard and be our best selves and do the best things that we can for humanity in the short time that we have on this earth.
Well, what a fantastic way to close. Dr. Siegwart, thank you so much for joining the podcast and this amazing conversation. And to our listeners, thank you for joining Learning from Leaders. We'll have some more information about Dr. Siegwart in the link in the bio, and we'll see you next time on the Learning from Leaders podcast.
Thanks for listening to Learning from Leaders. If you enjoyed this episode, we invite you to subscribe now for monthly episode drops so that you can continue to learn from experienced business and community leaders to help you drive meaningful change in your community.